All right, it is my distinct pleasure to welcome and introduce 5 incredible leaders from the field of fish health who are working across the country for the U.S. Fish and Wildlife Service. They're going to introduce themselves individually as they go along. This is a team presentation. We're super excited to hear the diverse perspectives that they all have. If you would like to see their full bios, though, I encourage you to check out our website where we have all that information posted, and I'll drop that into the chat here in just a second. But John, I think we're getting started with you. So you want to get going? We'll take off.
Thank you. It's a pleasure to be here. And on behalf of everybody else in the service, it's a pleasure to be asked to give an overview of the fish health program. So the Fish and Wildlife Service as an agency is very partner driven, and I would say that the fish health program within the service is even more so partner driven. And we just celebrated 150 years of conservation in the United States. Where I'm at, the Lamar Fish Health Center, it's in the Northeast region in central PA. We cover from Virginia to Maine. We have the Appalachians, we have the North Atlantic coast, including Chesapeake Bay, as well as the lower Great Lakes. Our staff is small, but we have a very diverse set of occupations in the program. And with that very diverse set of expertises, we share these expertises across the lab network, and we work very well with internal partners as well as external. So the genesis of the fish health program in the Fish and Wildlife Service was the need. In the past, there was all kind of unexplained problems in hatcheries, mortalities, and they needed to investigate when problems occurred. So therefore, the fish health program is part of the National Fish Hatchery System within the Fish and Wildlife Service. So depending on the watershed program, et cetera, we do what's called fish health inspections at our hatcheries, and which is getting them a clean or maybe not so clean, depending on the watershed, bill of health. And that's a statistical annual sampling, and it allows our hatcheries to transfer across state lines, stock in state waters, and receive eggs or fish from other locations. So besides the inspections, we also go back to diagnostic investigations, which was, as I said, our genesis. When problems arise, we're not looking for a known set of regulatory pathogens. We're looking for everything and anything that's causing the problem, be they infectious or environmental. We have a nice in-house capability for bacterial problems where we can do drug sensitivity testing and work with our aquatic animal drug approval program and our on-staff veterinary medical officers for quick, efficient, and FDA-approved therapies. Other services we provide is general information extension, kind of like county agents for our hatcheries. We provide training for our state partners. That can be formally at the National Conservation Training Center in West Virginia, a week-long class, very nice facility, pictured in the lower right. And we also have our state partners when they need to learn an assay or if they get a new piece of lab equipment, they come out for us for 2 or 3 days. We've had Maryland, Vermont, and West Virginia state folks come and get some training with us. So another program we do is the National Wild Fish Health Survey. That started in 1997 because whirling disease popped up in the Rocky Mountain region with no idea where it came from. So we decided it was needed to have a program to look at fish in the wild before there were problems, to get a survey of what's where. Some of the utilities we have within our region with our wild fish health money, we've helped out Maine with aquaculture escapees. These are adult domestic Atlantic salmon. We've helped out states when they want to bring wild trout into their captive breeding programs. In the Northeast, we're big on truck and transport of some anadromous fish because of dams. And the Wild Fish Health Survey has its own database, which has been developed and refined, and we have a soon-to-be online mapper in which you'll be able to click on various watersheds to find out what work was done and what the findings were. So we're working with wild fish, and we're in the Great Lakes region. And when VHS virus new strain popped up in the Great Lakes, it was a natural fit that when EPA was looking for proposals for projects to be done in the Great Lakes through the Great Lakes Restoration Initiative, we applied for a project titled Exotic and Emerging Fish Disease Surveillance, where we stepped up the wild survey and concentrated on the Great Lakes Basin, looking for VHS type 4B virus, which caused widespread mass mortalities when it first appeared. 37 species were affected. And so we're looking for that and several other new agents. We're all familiar with aquatic invasive species, plants and fish and mussels invading the Great Lakes, but microbes are also coming in. So another thing, when the VHS virus first appeared, there was a moratorium, an emergency order on no fish transfers out of any Great Lakes states. So our two states, New York and Pennsylvania, they really wanted to piggyback or partner with the Wild Fish Health Survey for us to look throughout the state, because so much of their state is not in the Great Lakes drainage. They wanted us to help them demonstrate freedom of VHS virus. Our hatcheries also got on Great Lakes Restoration Initiative funds to take up a new project, raising the traditional forage fish of the Great Lakes, lake herring and lake bloater. They were really depressed, extirpated, and alewife have replaced them. Alewife is not as suitable, both because of thymidase issues and because they're not as deep a fish as lake trout are. So they all started raising two service hatcheries and one USGS hatchery are raising them. And that brings us Fish Health to screen the adults, since they're wild adults. There was no propagation of these before this program. And then we give them several other tests before we put them back out, because two of our hatcheries are not in the Great Lakes drainage when going back into the Great Lakes drainage. Curiosity findings through the Wild Fish Health Survey. Here's a case of finding a large snakehead inside of a largemouth bass. So it's a case of a predator, a nationally renowned, feared predator becoming the prey. It's a fish eat fish world. So we are focusing on VHS virus, and it's not making the headlines anymore, but we still do see it from time to time. We do see it in times without disease. And we also did pick it up in a disease case 2 or 3 years ago. We got a phone call from a small university right on Lake Erie, saw some distressed fish and wanted to send them. We suspected VHS virus, and it was. We've also found some of our other traditional diseases where we weren't really expecting them. Another service we provide to our hatchery program is biosecurity. It's proactive to keep diseases out, controlling vectors. Picture right here on the upper left is our flagship lake trout hatchery in northwestern Pennsylvania. It's right in the left side of that picture is a huge dam with a constant spray of water, and it's in the endemic area for IPN virus. And in 2005, they came down with IPN virus, and they suspect it came through birds. Those green little covers were more white than green because of the birds. And we disinfected, we renovated the hatchery, new pipes, new aeration building, but more importantly, we addressed the bird issue. And since 2012, it got back online. We have not had IPN virus found. The other service we provide is risk management. We want to reduce the risk of finding a disease. The case of our Atlantic salmon, they come back from the North Atlantic, and the Penobscot River in Maine is the last river with a large run of Atlantic salmon, which this strain is listed as endangered now. And they go up to the Milford Dam. It's where we're able to get our hands on them, collect them. And Craig Brook Hatchery is our facility that holds them, spawns them, and releases the fry back. Unfortunately, when the salmon are on their migration, there are marine aquaculture net pen sites they have to pass by. From time to time, those commercial sites come down with a disease of Atlantic salmon called ISA, infectious salmon anemia. And it's kind of a lot of finger pointing in the early years, whether the wild salmon brought this into the aquaculture setting, or if the aquaculture setting is putting the wild fish at risk. The more we learned about it, there are 2 strains of this virus, North American strain, which probably was there ahead of time, and it's non-pathogenic. And then there's the European strain, which is pathogenic, which may have been brought in early on when these net pens started. They were using salmon from Norway because Norway is well advanced in the marine net pen Atlantic salmon industry. So our hatchery dealing with these salmon is Craig Brook. It's the first salmon hatchery in the United States, started in 1871. However, it has gone through major renovation, modernization. It has 7 isolation quarantine areas. It has UV filtered or microscreen and sand filtered water and UV sterilized water. And the Penobscot portion is right over here. There's 4 little sweet pools that are covered. And the straight building is 5 isolation bays. I'll talk about in a little bit. It'd be really nice when you're dealing with wild broodstock that are at risk of a disease to be spawning your fish by pairs or by some similar 4 fish or 6 fish, where you could track those fish and their eggs and egg jars. But the limitations did not let that be part of our design. So we have heath stacks where the water flows from one to another, to another, to another. And therefore we have a lot of risk should something pop in the upper trays. Fortunately, when you work with partners come to you. And in the University of Maine and USGS came to us, and NOAA Fisheries want to know if we could participate in a blind study with 3 labs, a Canadian lab, the state of Maine lab, a private lab, and our lab, where we'd compare 3 different tests and 2 different tissue types. And the tissue types were blood and the traditional lethal kidney spleen. Well, it's fortunately that it was no surprise that DNA testing, PCR, was the most sensitive test. But what was nice surprise was blood was as good, or maybe a little better, but not statistically better, but it was as good as the lethal sample. So we now had a non-lethal tool to screen our fish prior to spawning. So the fish are captured at Milford Dam, brought to these isolation bays at the hatchery. Each day will be a different bay. There they are pit tagged for individual ID, fin clip for genetics for genetic spawning later in the fall. And then blood is drawn and sent to our lab for ISA screening. So we test the traditional cell culture test for ISA virus. However, that takes 28 days. We still do it side by side because it is the gold standard for this, for most fish viruses. And the PCR to DNA, we can get a 2-day turnaround, like 1-day extraction and 1-day run. So the fish have individual IDs. Should we have a hit or a positive, basically a suspect, we can go back scan them, and find that fish and get it back out to retest. And if it's, next step is to sequence the virus that we find in that fish to see if it's the pathogenic or the non-pathogenic. We're fortunately co-located at the Fish Tech Center, which has the genetics lab here. So we work very well hand in hand with them. So the ultimate goal is if they're negative, they come in to these 4 Swedish pools, which is really a Craigbrook Salmon Spa. The Penobscot River gets quite warm as the months go on before spawning. We're here, we keep the fish cool, dark, and happy. None of them are feeding while they're here because they're on their spawning run. So here's how it works. On the left is the river and the dam. Fish are collected. Some fish are passed over to spawn on their own. That's the ultimate goal at the end of the restoration recovery. But so the fish are brought to these 5 isolation bays and they're screened. If we look at number 2 here, if they all test negative, they get to come into the Swedish pools and be held till October. Fish are coming in right now. We've got bloods today and tomorrow. So they're coming in May, June, July, and they spawn in October. But if we get a hit, then we have to sequence it. And its cohorts can come into the hatchery, the ones that tested negative, and we pull this guy out and we determine whether or not he's pathogenic or non-pathogenic. If he's if he's non-pathogenic, he gets transported above the dam and put back in. We don't have a problem putting non-pathogenic but positive fish back out because a lot of his cohorts were just simply passed untested, and a portion of those are non-pathogenic carriers. We don't want to bring it in the hatchery even though it's not pathogenic because we kind of see this in a 3 to 5% range in the population over each year. And we're afraid if we bring it into the hatchery, we could biomagnify its incidence in the hatchery and change next or 4 years later what the run looks like. So if we get a positive fish, then all of its cohorts are held for 28 days and retested. And the positive fish gets put down, extensive sampling done, and sent to USDA Ames Iowa lab since it's a nationally reportable disease. It would be devastating to the hatchery should we have a nationally reporting disease in the hatchery proper, but the use of these isolation bays and good biosecurity between allows the hatchery to operate. And then the ultimate goal is to spawn these fish and hatch them out and keep the population going. Turn it over to Lacey.
Yes, hello everyone. I'm Lacey Hopper. I am the project leader at the Bozeman Fish Health Center up here in Bozeman, Montana. I apologize, there's yard work going on at my lab right now, right behind my window. So if you hear, mowers and such, hopefully it won't affect the sound too much. So a slide. So a little history about the Bozeman Fish Health Center. We were originally named the Fish Disease Control Center and established in 1974 at Fort Morgan, Colorado. You can see from this old booklet that I found, our region used to have 2 additional states there, Iowa and Missouri. So over time, we lost those regions and now have just the 8. We moved to Bozeman in 1996 and were renamed the Bozeman Fish Health Center. The hope for this move was to increase partnerships since it would be near Montana State University. It would be more, there would be more scientific advancement due to the location to the Montana State, more research opportunity, and of course, more centrally located within our region. Our current staff is 4 full-time employees and 1 part-time. And we just recently hired an aquatic veterinarian, and we look forward to him joining our team. Slide. Okay, this is the U.S. Fish and Wildlife Service Region 6, or Mountain Prairie region that we cover. We cover 8 states. It's a very large geographic region. It keeps us very busy. We provide regular services, so not just once in a while. These are folks that we work with regularly. We have 12 national fish hatcheries, including the largest primary rainbow trout broodstock facility, Innis National Fish Hatchery. We have 1 tribal fish hatchery that we go and provide inspection services to. We have a tech center, the Bozeman Fish Technology Center, that we work regularly with. We have 7 fish and wildlife conservation offices, or FWCOs as we call them. We go out and sample wild fish. We work on special projects, pathogen monitoring with those Fish and Wildlife Service partners. We work with partners from all 8 of our states on fish health type activities or testing, AIS needs that states may have. We're very active with our state partners and very supportive of our state partners. We work with multiple tribes within our region, wild fish survey, if they have fish kills within a tribal reservation, that sort of thing. We are lucky enough to work with 3 national parks. We get to go into Yellowstone, Glacier, and Rocky Mountain National Park. So those are some of our favorite wild fish health samples. And we work closely with the National Park Service on those. We also work with 4 main USGS offices on various collaborative projects and any other needs. Sometimes we share equipment. We're fortunate enough to have a great USGS partnership here in Bozeman. And then we also provide services to fish and wildlife nonprofits. We have a Wolf and Grizzly Discovery Center who just down by in West Yellowstone that put in a living stream for Arctic grayling and Yellowstone cutthroat. So we've been working with them getting that stream up and going. Slide. So this is just some pictures of some of our priority species. Our list of species that we provide services to is quite large. We work with cool, cold, and warm water species within our region. So some of our priorities are, of course, West Slope cutthroat, Yellowstone cutthroat, the Wyoming toad, endangered Wyoming toad. We have a national fish hatchery, Creston National Fish Hatchery, who gets bull trout eggs in. They are the only hatchery that I know of that can artificially rear bull trout within their isolation buildings. So Arctic grayling, paddlefish, pallid sturgeon, shovel-nosed sturgeon, all of these are priority species for our region. Next slide. So some of the services that we provide are, the same as John's lab and other fish health centers. We provide the standard inspections, diagnostics when those are needed. We go out and we do national wild fish health surveys and monitoring. We respond to fish kills within our region. Other things that we do are applied research, special projects. So we do several population genetics projects with our Kendall warm spring dace, our Wyoming toad, and multiple other species within our region. We are trained and respond to natural resource damage assessment. So oil spills, we've responded to 3 of those within our region. And that requires a lot of special chain of command, sampling protocols, a lot of coordination, a lot of archiving, documentation, because of course those can go to court. We are now providing flow cytometry testing here for triploids. That came because of our Innis National Fish Hatchery. provides triploid eggs. So we're doing that testing now for them and other state partners that reach out to us that have that need. We also have an invasive carp eDNA testing program here now. We provide eDNA testing for bighead and silver carp within the upper Missouri River Basin. We also monitor pathogens within some of our facilities for, like a source of an infection somewhere. So we've been successful with that. We are trained to provide facility AIS inspections. We were trained by our Montana state partners and work with them on that. And then we provide chytrid testing for wild toads and amphibians, as well as the toads that are reared at our Saratoga and Leadville National Fish Hatcheries. So these are just some photos of us doing some fun stuff. Slide. Again, some of our field, where we're out filtering water for eDNA. We're at one in the middle is Rick Cordes. He's filtering water at our Hotchkiss National Fish Hatchery for pathogen monitoring in their spring. We've got Rocky Mountain National Park sampling, sturgeon. We do a lot of sturgeon work and sturgeon health within our region. Next slide. Some of our regional issues include, I'm sure a lot of you have heard about the Tetracapsuloides bryolsalmone massive fish kill. That was in 2016 on the Yellowstone River. It mainly affected adult mountain whitefish. The losses were in the tens of thousands, and we worked with our Montana state partners to determine that it was T. bryolsalmone that had caused that massive fish kill. It was a drought year. It was a perfect storm for that parasite. So we're still looking into that. You can see some of the histo slides of that parasite from that fish kill. Other issues we deal with are, of course, invasive carp monitoring, helping our partners monitor that. Security and infrastructure. Our region has limited budgets, so putting covers over everything, and a lot of our hatcheries are very old. So trying to keep up with biosecurity and updating infrastructure to keep pathogens and AIS out you have to be creative sometimes to try to keep things within budget. So again, keeping pathogens such as whirling disease and New Zealand mud snails out of our hatcheries. We have, probably like most folks, gas supersaturation issues at some of our hatcheries and out in wild fish as well. We've seen that. We're currently investigating and working with Montana Fish, Wildlife, and Parks to investigate what they've kind of termed cheese grater head. It's these lesions on the tops of the heads that we're seeing in multiple rivers in mountain whitefish and brown trout. So we're working with other labs and with the state partners on that. We have cutthroat trout fry. We found that they can be difficult to rear and get them up on feed. So we're working with that on early survival of cutthroat trout. And then every now and then we get Missouri River sturgeon iridovirus in our pallid sturgeon, or endangered pallid sturgeons. So we are dealing with that at times. And if we get it in a hatchery, trying to, figure out how we can avoid it next time, that sort of thing. Next slide. So one thing I wanted to throw in and talk about is if folks are interested in joining the Fish and Wildlife Service. I've been asked by folks, hey, how do we start working at your lab or one of the fish health centers? And I wanted to throw this out there for any students or folks that might be interested in joining our service team. I guess one thing would be willing to volunteer. We always are accepting and needing volunteers, and that could potentially get your foot in the door. Look into student opportunities. They're out there. I provided the website here. They're, for high school students, college students. There's definite opportunities out there for folks to go to different labs or different Fish and Wildlife Service facilities to learn more. Be willing to move. A lot of us in the Fish and Wildlife Service have moved to multiple facilities, and it's not necessarily a bad thing. You learn things from each of the facilities, each of the different regions. Ask questions. Never be afraid to ask questions. We love to answer questions. Find a mentor. I did. I started at the Idaho Fish Health Center, and all of the folks there at the Idaho Fish Health Center were mentors for me. And then most of all, be passionate. Be passionate about what we all do in aquatic animal conservation and recovery. And that's it. Thanks, everybody.
Hi, so I'm Trista Becker, and I'm the supervisor of the fish health unit here in the Southwest. We are located at, you may have heard the name, Southwestern Native Aquatic Resources Recovery Center, or SNARC. And slide. So we are in the region 2, which is the Southwest region covering Arizona, New Mexico, Texas, and Oklahoma. So wide diversity of habitat types. We're in the desert in the southeast corner of New Mexico, a little place called Dexter. Next slide. And we are co-located, so the center is made up of the Fish Technology Center here, and the Fish Health Unit, and a National Fish Hatchery. We are a Tier 1 accredited lab, like most of the other fish health labs in Fish and Wildlife Service now. We do the standard gamut of virology, bacteriology, parasitology, and we do our own molecular confirmations for some pathogen detections. Majority of the work that we do is hatchery inspections for the region. Our national fish hatcheries, we have a tribal hatchery in New Mexico that we do inspections for, and the Oklahoma state fish hatcheries as well. We also do diagnostics when needed, mostly just for the federal fish hatcheries. And since I'm a veterinarian, I can do prescriptions. We can prescribe treatments for as needed. And then as time allows, we do wild fish health survey work around the region as well. And that's just a photo of the administrative building here on the center, the front of the tech center building. Slide. So we have currently 5 of us total. We do have 1 vacant term position right now. He took a job up in Abernathy, Washington, so he was able to get on permanent with Fish and Wildlife there. And so in the top left, you'll see Hussein, and then Jason actually is in the bottom right. And I was able to give him the 20-year service award last year, which was a big honor. And then you see Dave in the top right, and then Matt working in the molecular lab in the bottom. And so our staff, we all do the inspection work, and in the lab, the sections are broken up where we have 2 people per section roughly. So Dave and Hussein are currently working in virology, and Jason and Matt are working in BACT, and then Matt does our molecular work as well right now. And then, like I mentioned, I'm a veterinarian, but my title is actually supervisory microbiologist. And then the positions are in our lab are specialized fish health positions, but they're classified under the fish biologist series. So if you wanted to find out more about the qualifications for those positions, you would go onto the OPM site and look for the fish biologist series. Next slide. So we do majority, like I said, of our work on the hatcheries and doing inspections for fish movements. And we do all kinds of different fish, large and small, anything from tiny little darters to very, very large catfish. Next slide. And again, just some more photos of people doing work out in the field, and Matt looking for Asian tapeworm there in the parking lot. Next slide. So most of our hatcheries, the overall goal is to support the endangered species programs or the endangered species within our region. And some of them have stocking programs that with the goal of recovery, and some of the main species that the hatcheries do for those are the razorback sucker, Rio Grande silvery minnow, bunny-tailed chub, and Gila trout. In the top right, you'll see the Rio Grande silvery minnow, and so those are a small fish in the Rio Grande, but very important. And we do have quite a bit of sport fish as well, especially with the state hatcheries that we help out. So, you know, all the gamut of species from trout to catfish and carp and sunfish and all the hybrids that they like to make in between. We do have, the last count I saw was 22 species in Refugia within the region, and I believe 14 of those are here on station in Dexter. And most of them are fish species. We do have some toads in Texas and aquatic salamanders in Texas as well. And then the alligator snapping turtle, there's a Head Start program in Oklahoma. And we do work with partners for around Texas, some of the zoos for the Houston Toad program. The fountain darter, one thing that we do for the fountain darter is when they collect from the wild to bring them into quarantine, and so we do testing for those. And one thing that's been kind of an issue in recent years has been LMBV, especially since it's kind of all over Texas. So it's something that we are looking at right now. And again, we have partners from the states and tribes, the refuges even, and some of the zoos, especially in Texas. Next slide. And so Refugia, like I mentioned, we have fish in Refugia. And so basically fish are brought in and held kind of as backup when there's low population numbers in the wild, or if the habitat is under threat. So one thing that's recurring and actually going on right now in New Mexico is fires. And so when fish are salvaged out from in front of a fire front, or if there's issues with water drying up in the habitat, then fish will be brought in. And then if they're brought into a station, then we will need to sample and test for the routine pathogens. And the Gila trout are heavily impacted by fire in New Mexico. They have been previously, and so right now the black fire is kind of a threat, ongoing threat in New Mexico in the southwest corner. And then our Mora hatchery, we were recently faced with threats because of the hermit fire up in the northern part of the state. So next slide. And I think I counted 49 species, and this list is growing all the time. This is all the different species we work with or have worked with. And you probably noticed that most of them are threatened and endangered. So while we look for the standard list of pathogens that everyone looks for, we don't always necessarily know what a detection means for that species. Some of these species, we know very little about. And even though we do occasionally, like I mentioned with LMBV on the fountain darter, get detections, we don't necessarily fully understand yet what that means for that species in particular. That's it for my section.
Hello, everybody. My name is Ken Phillips, and I'm the project leader at the La Crosse Fish Health Center, which is located in Onalaska, Wisconsin. And we, along with the Whitney Genetics Laboratory and the La Crosse Fish and Wildlife Conservation Office, make up the Midwest Fisheries Center. So like it's been mentioned, like the other fish health centers, we provide inspection and diagnostic services to the 6 national fish hatcheries and 7 tribal fish hatcheries here in the Midwest region, as well as provide technical assistance to some of our state partners and USGS partners. The focus is pretty diverse for the program, fisheries program in our region, everything from Great Lakes restoration of trout and gorgonids to rainbow trout mitigation to mussel restoration. So it gives us a lot of opportunities for the staff at the center to work with a diverse group of species and really work with a lot of different novel species of bacteria, parasites, and viruses. So slide. So one of those is kind of emerging bacteria that we've had an opportunity to deal with is some lactic acid bacteria in the region, going back to fall of 2017 after spawning of brook trout at the Iron River National Fish Hatchery, which is in the upper left. The Iron River National Fish Hatchery rears both brook trout and lake trout, and they have both broodstock, which is held in the far left building, and then they also have production for both species in the building on the far right. So after spawning of their brook trout in the fall of 2017, in early 2018, they reported mortality in their broodstock. And when they would open up the fish, they would notice egg retention, as well as a white fibrous material around the cardiac tissue and some ascites fluid. And some of the egg retention actually was the skein was still completely intact with those eggs. So working through doing a diagnostic on those fish, we identified the pathogen as Vagococcus salmoninarum, which is a lactic acid bacteria, gram positive. And it's not really new. It had been, isolated as early as the late 1960s in the Western US. But it's really kind of had been a more obscure pathogen. We tried, treating with Aquaflor, but it was not very effective. It would reduce the mortalities, but as soon as the treatment stopped, the mortalities would return. Over the course of 2018, the hatchery lost probably close to 2/3 of their brook trout broodstock. So the decision was made to cull the broodstock. They just couldn't sustain it, which gave my staff an opportunity to really study the pathogen while looking at the various tissues that served as reservoirs as well as vertical transmission. So we credit to especially Isaac Standish on my staff for kind of writing the book, I like to say, on Vagococcus salmoninarum. We also noticed, though, through screening for Vagococcus, we also noticed another gram-positive bacteria that was very similar, and we identified it as Carnobacterium Multimaticum, which is another lactic acid bacteria, very similar. And that actually continued causing some mortality issues at Iron River. Vagococcus kind of, we ended up culling the broodstock, and Vagococcus was no longer a problem, but the Carnobacterium continued to cause a problem at the hatchery. And ultimately, we developed a duplex qPCR for both Carnobacterium and Vagococcus, which allows us to basically, instead of doing 2 separate assays to see if we have Carnobacterium or Vagococcus, we can hit them all at once. And then a unique thing about this is that 2 of our tribal partners, the Red Cliff Tribal Fish Hatchery and the Kootenai Bay Tribal Fish Hatchery, all of these hatcheries are in the Lake Superior Basin, also had Vagococcus. Red Cliff, basically had significant losses of their brook trout broodstock, and whereas Kootenai Bay did not. So part of the study with these different pathogens, different strains, is we are working with the Whitney Genetics Lab to sequence the genome of the Vagococcus and Cardinal bacterium. And the sequence is actually done, and we're working on finishing up the analysis, hopefully identifying some genes for antibiotic resistance and virulence in order to maybe see why we see some of the different things with these strains. Slide. Working with the diverse species that we work with, we also get an opportunity to isolate and further describe some novel virus species, some of which are shown here. Slide. And then also, we also have a work with quite a few novel parasites. Becky Lacey was a former project leader at the Fish Health Center, and I would consider her a world-class fish parasitologist. And she passed a lot of her knowledge on to several of us at the Fish Health Center. And one staff member, Eric Leese, is especially taken a passion for fish parasitology. And he really studies a lot, and he's often receives requests from throughout the United States and the world to help identifying parasites, some of which are often novel. One of those was the Gyrodactylus malus, was actually from working with individuals from the Czech Republic. And actually those individuals are planning to visit our lab next month to work with Eric and kind of get some experience here. As well, one other parasite, and I'll touch on a little bit later, is the Microsporidium Clinch Eye, which is from pheasant shell mussels, part of a mussel project that several staff at the Fish Health Center are working on. So slide. So mussel investigations, as most of you probably know, little is known about the pathogens and diseases of freshwater mussels. So several members of my staff, Dr. Isaac Standish, Eric Leese, and Sarah Gazicki, are part of a team with investigators from the Upper Midwest Environmental Sciences Center and the National Wildlife Health Center in La Crosse and Madison, respectively, both USGS labs, and the University of Wisconsin. These mussel investigations began with some die-offs in the Clinch River, which is in Virginia and Tennessee. But from there, this team kind of got word spread about them, and they also started receiving information about die-offs from the Embarrass River in Wisconsin for two species, the mucket and pink heel splitter. The Crooked River in Oregon with the Western Ridge mussel, the Keelis River in Washington with the Western pearl shell, and then the Huron River most recently in Michigan with some muckets. Doing this work, they've developed where they can use hemolymph from the mussels, which is very important. That's a non-lethal sampling method, and many of these mussels are threatened or endangered. And through this work, they've identified numerous potential bacterial, viral, and parasitic pathogens, working on developing some molecular assays for these pathogens. And then slide. One of those pathogens that has been, novel pathogens that has been discovered from some of this work is a Microsporidium, the Microsporidium Clinch Eye. It was found in developing oocytes of pheasant shell mussels from the Clinch River. And so it's really interesting. I know that these oocytes would be completely filled with this Microsporidium, leaving no viable oocytes in these mussels, which obviously would be very detrimental to the pheasant shell population. So with that, I think I'm done, and we'll pass the baton to Guppy.
Thanks, Ken, and hi, everyone. I'm Guppy Blair, the branch chief of the Aquatic Animal Drug Approval Partnership Program, or ADAP. However, before I talk more about ADAP, I'd like to just give a very brief overview of the Idaho Fish Health Center, where I was stationed a little over 20 years. I do have to point out, though, that I'm only going to talk about my experiences at the Idaho Fish Health Center, since things have changed quite a bit since then. And we did provide support for national fish hatcheries and partners. And similar to the other fish health centers, we did fish health exams, but ours were primarily on Pacific salmonids. So we provided diagnostics, inspections for movement, testing at spawning for returning adults, pre-release exams, and monitoring. And at that time, our laboratory testing was very similar to the other fish health centers that I've already spoken today. However, since then, the approach has changed to where they're contracting all of the laboratory testing out, and they're focusing on field work and disease management. So that's a little bit different. Next slide, please. All right, and I just wanted to give an idea of the different pathogens that we might see and pick up, detect at different times in different places, pathogens and diseases. So I won't go into each one of these at all, but I did want to point out the first one on the virus list there, infectious hematopoietic necrosis virus. We did quite a bit of testing and studies, worked closely with the Western Fisheries Research Center within USGS, Gail Carafe and Rachel Breda on genotyping and looking at the epidemiology of the genotype that the returning adults were bringing back and the timing of when of the different genotypes and how that affected the fish in the hatchery. So we'll go moving on then to the next slide. So some of our primary partners were the Nezburg tribe and the Kootenai tribe. Nezburg have a lot of things going on with their own hatchery and associated acclimation sites. They have operation of the Kuski National Fish Hatchery. They have fall Chinook acclimation sites, coho program, and lamprey program. So a lot of things that we helped them out with at that time. And then the Kootenai tribe have the endangered Kootenai white sturgeon. We did a lot of testing for white sturgeon norovirus, and then also worked with them on their burbot program. Next slide, please. All right, so to change gears a little bit, as I said, I wanted to talk a little bit about the Aquatic Animal Drug Approval Partnership, or ADAP program, where I am stationed now. Next slide. And for ADAP, our mission is a little bit different than the fish health centers. We do assist fish health and fish culture, but primarily our mission is to obtain FDA's approval of safe and effective new medications for use in aquaculture and fisheries management. Next slide. And so who are we exactly? Well, we're a national program administered out of headquarters in Washington, D.C. However, we're located in Bozeman, Montana at the Bozeman Fish Technology Center. Currently, we have a staff of 6 employees, including myself, so it's a very small team. And we're unique. We're the only program in the Fish and Wildlife Service that is 100% dedicated to aquatic species drug approval efforts. Next slide. And so what we do are several different things. One is to conduct research to support new animal drug approvals, and I'll talk a little bit more about that later. We also implement our national Inad, or Investigational New Animal Drug Program, and I'll talk about a little more about that one as well. We disseminate aquatic animal drug use information to try to keep everyone up to date on as that changes. And this definitely we can't accomplish alone. So ADAP holds a partner-based, nationally coordinated effort of federal, state, tribal, academic, and private sector partners. And we highly, highly value these partnerships because, again, we couldn't do this on our own. Next slide. So steps for drug approval, just to give you an idea of that it is very difficult. It's not easy. There's 5 major technical sections that must be fulfilled. And it's the same as for terrestrial animals. So aquatic animals don't get any shortcuts. ADAP primarily works with the top 2 sections listed there, target animal safety and effectiveness. And what they are, just real briefly, is target animal safety. It makes sure that the drug is safe to the target animal species. Effectiveness is to ensure that the drug does work in that target animal species when it is used at the proposed label claim. And then human food safety. This is where food products made from the treated animals must be safe for people to eat. The chemical manufacturing controls section, this is where it involves the ingredient used to make the product, how it's made, packaged, and stored. And then finally, environmental impact is considering how the environment will be affected with an approval of a new animal drug. So each one of these sections can take multiple studies. Next slide, please. So that brings us to our research program, where we do conduct pivotal efficacy and safety studies. And this has to be done at the level that FDA requires. So there's has to involve controls, dose verification, blinding, good laboratory practices, quality assurance level, those types of things. And again, these are all needed to complete the technical sections for the drug approvals. And then we either conduct the studies on site at the Bozeman Fish Technology Center or at other facilities across the country, because FDA does prefer that the efficacy studies be done in the field with natural infections. Next slide. And then I want to talk also about our investigational new animal drug program, or Inads. This is really a temporary solution. And what they are and why they're important is that they do allow the use of unapproved aquatic animal drugs as they're going through the drug approval pipeline. So they're not approved yet, but we're working on them and making progress. And currently, ADAP holds 18 different Inads that are available for participants to be able to use. It's all under very controlled situations and how they can be used. And then, but they do cover therapeutics, such as for disease and parasites, spawning aids, anesthetics, and marking aids. Next slide. So I just want to finish up with the best parts of the job for ADAP. It's the resulting approved drugs that can be used to help keep fish healthy and to facilitate safe handling, spawning, marking, and other fisheries management activities. And then I think we'll all agree that it's really the people that we work with, collaborating with numerous partners and colleagues in support of aquatic animal recovery, conservation, and recreation makes the job rewarding and very enjoyable to be able to work and in partnership with so many different people. So next slide. All right, just want to thank you all for attending, and hopefully that gives you an idea of what fish health can look like as a career in the U.S. Fish and Wildlife Service. And if we have time, I'd like to open up for any questions.
All right, thank you everybody for those perspectives and experiences. There's a lot going on. It's pretty incredible what the service is up to. We're right at time, but I can hang out a little bit longer if folks want to drop a question or two in the chat, or feel free to unmute yourself and ask somebody directly. Any questions out there? All right, if not,
I'll chime in, Nick.
Go ahead, Matt.
If everyone else is going to let me be the one, I'll hold on one more minute. Lacey, this one's for you. I'm just curious, the invasive carp eDNA testing that y'all run, is that a LAMP assay? Is that like a TACMAN PCR assay? What level of sensitivity is it? Are there protocols out there that are available for folks if we want to try and apply those to other potential invasive organisms?
Yes, so it is, Yeah, qPCR assay. We follow the protocols from the U.S. Fish and Wildlife Service Whitney Genetics Lab. Their QAP, QAPP, and so we have worked extensively with that lab to get our lab up and running at a much smaller scale than Whitney Genetics Lab, but they have been fantastic to work with. We were asked by our region if we could do this and if we had the capability, because Whitney Genetics Lab had so many samples coming in, they weren't able to take on our regional samples. And so we don't really have a service genetics lab within our region. So they kind of came to us with that. And so we quickly got with the Whitney Genetics Lab, and they provided us with guidance. We actually took a storage closet that we have in the lab, and we turned it into an eDNA extraction lab. So we bought, additional equipment that's solely used for eDNA. We moved some of our equipment around so that we would have individualized areas to do each part of, eDNA processing, so that, we didn't run our standard or regular confirmatory PCRs in the same kind of extractions in the same areas as eDNA. So it's taken a lot of maneuvering and trying to be creative in making sure that we're, reducing contamination risks. So I would look into the Fish and Wildlife Service QAP for those protocols.
Okay, excellent. Thanks.
Yeah, you're welcome. Thank you for the question.
Tom, I see your hand up.
Yeah, I just wanted to echo what Nick said. Thank you all. That was awesome. And it's totally impressive to see all the different things that you all have going on. I'm wondering how much you all coordinate amongst the different fish health centers. Like, are there any pretty large scale projects that you all have been sort of tackling as a group, or that sort of is like a situational type of thing when something arises? I guess I'm just wondering about coordination amongst centers, what that looks like.
I'll jump in first, but I know some other labs have doubled up on projects, but at Lamar, we, back when we had a first occurrence of spring viremia carp in Virginia, or North Carolina and Virginia, we teamed up with the Warm Springs lab and did an extensive week of sampling and actually processing, because back then they had the mobile lab, like a little RV. And we worked with North Carolina and Virginia fisheries folks to get as many site print-ins as we could to see if it was out and about. It was initially at a goldfish farm. And then there was the VHS, we partnered with Crosse, and we were helping Lamar went over to Ohio. I think we were looking at some wild broodstock for VHS. I don't know if anybody else wants to add.
Recently, since there's shortages in some of the labs, I've sent staff. Devin's not on here, but the Southeast region, I just sent a couple people to do details down there. And we've been coordinating on the accreditation, I think communicating with each other on lab accreditation lately.
And I can jump in too, like, I've worked with, like John mentioned, with his lab, I've worked with Lacey, as, certain projects where overlap our regions. And then we do share expertise, quite a bit across our regions, sharing information. For instance, at duplex qPCR assay, we've shared that with all of our other fish health centers, made it available to them. So that, they can do that work for the Vegacoccus and Carnobacterium. And that's just some of the examples of the stuff that we do.
Yeah, I wanted to jump in and say that we're very supportive of each other. If one of our cell lines crash, we immediately know that we can call each other, and the others will get, a flask out as soon as possible, maybe even 3 flasks if we can send them. We send each other to each other's labs to help when needed. There was a recent fish kill in Nevada, and Scott Flitt's lab at Cal Nevada is extremely understaffed. And so we stepped in and took on those samples for that fish kill, even though it wasn't in our region. We just wanted to help out. And so, yeah, I'd say we're very supportive of each other and jump in whenever anyone needs anything.
That's great. Thank you. Yeah, definitely a very potent, powerful network. So cool to hear. Thanks.
We got one last question here. It's in the chat. An undergraduate student is tuning in from Nigeria right now, and they're curious if there might be international student internship opportunities with the service.
Boy, I don't know that answer right off the top of my head. I'm sure there are. Maybe other folks can jump in that might have had an experience with an international student. But again, I would say go to, our website and just check it out and see, if there might be more information on that on the Fish and Wildlife Service website for student opportunities. Unless one of the other lab has had experience.
Yeah, this is John at Lamar. We run our internships. We haven't had that many, but we run them as volunteers. So that would kind of make it tough for an international student. But if the interest is there, I think any fish health center would take a volunteer that work as an intern.
Great. Well, that's all the time we have for today. Thank you very much to the 5 of you for presenting. That was an excellent series of talks.